We're on. Hi, everybody. Whatever. Okay. So we're talking about some of the cell biology going on here and the difference between prokaryotes and eukaryotes. And, there's some very critical differences between the two different systems, the two different types of cells. And this is important because it allows us to have really functional antibiotics, but at the same time, because of this, there's a huge amount of similarity. Bacteria, not from a disease perspective, but from a research perspective, have been a brilliant model system for understanding genetics and mutagenesis and protein structure and gene expression and tons of nerdy molecular genetic things. And one of the cool things I think it's important to keep in mind is that any gene can be expressed between the two different systems. I mean, there's a little bit of tweaking you have to do, but the whole principle behind modern molecular biology and modern molecular pharmacology is the fact that you can take a human gene, put it into a bacterium and express it. So, we're currently working on building a bacterium that will express the major protein, the major envelope protein from Zika virus. And let's see, I think it is the Johnson & Johnson vaccine currently up and running, that they are injecting a bit of the spike protein into people. And the difference is that Moderna and Pfizer, they are injecting the RNA that'll code for the spike protein. The more traditional way of doing vaccines for the longest time is to actually put in either an attenuated virus, which is what the AstraZeneca vaccine is. It's a modified adenovirus, which is not pathogenic, that expresses a bit of the spike protein. Or you just inject the stupid protein into somebody, and you make that protein probably in bacteria, in a fermentation system. So you put the gene for whatever protein you want into the bacteria, you have them express it, and you produce it in a vat like the size of a Bush brewery or something. You collect, if you're clever, you can actually have the protein being secreted into the media. You collect the media, and you got a rather tedious purification process, but there are some genetic tricks that you can build little, it looks like a little knob on the back end of the protein that you can grab with a system. Grab that protein that you want, wash everything else out, and then release your protein, kind of let it make the handle let go, and produce a very pure protein. And that's probably what they're doing. So this is because bacteria and eukaryotes, we share the same genetic code. So that's a pretty cool thing. And what I had finished up on when I was looking at the lecture from last fall was the notion that a good idea would be to build a list like this and to write it out so you are incorporating it into your brain better than just by reading it. The differences between prokaryotes and eukaryotes. And I thought there was a way, you used to be able to turn that light out especially, but I'm not sure you can. I'm going to take 15 seconds out of my lecture time to fiddle with that for a second. I bet it's not here, but it might be. That light. Thank you, the very front. Didn't used to always be on. All right, regardless. And that's the switch for the broken screen. All right, so making yourself a list is a useful thing to do, because this is a really important thing that you'll see questions on, let's face it. All right, we finished up then with the similarities. You can always talk about the differences, but the similarities. We've got the cell membrane, ribosomes, the cytoplasm, DNA, And protein synthesis, I mean, it is absolutely the same thing, and in fact, the mechanics of it were discovered in bacteria. And so that leads to our simple dogma that is the watchword of molecular geneticists. All right, so now, back to actual microbiology stuff. One of the key things in microbiology as a general study is being able to distinguish different bacteria types. And so we'll be playing around with this in the lab. We have our unknown project in which you will get a bacterium, and then you have to actually figure out what is the species, what is the type of bacteria. And this is critical because certain antibiotics are really effective on one class of bacteria and completely ineffective on another class of bacteria. There are bacteria that have very specific nutritional requirements. There are bacteria that cause very specific diseases, and knowing what bacteria you got is important. And knowing the kinds of environments they can grow in is also really critical. We have some bacteria that can grow on the cell surface, or our skin surface, and we have other bacteria that are going to grow only in areas that don't have a whole lot of oxygen, so like a deep puncture wound, or deep inside your digestive system in the colon where there's not a lot of oxygen going on. And knowing the different kinds of bacteria becomes a really critical thing. And so we got a whole range of bacterial types. And because this is focused primarily on medically healthcare related relevant bacteria, happily there's a whole class of bacteria and a bunch of them that are not going to be part of our picture because they're not pathogenic, they don't grow in people, you don't have to worry about them. But we talk a little bit about them. So we've got pretty much bacteria can be found everywhere on the planet. There is no place on our Earth that you're you take samples and not find bacteria. I mean, short of probably actual lava. We find bacteria in boiling water vents in Yellowstone, at what are known as black smokers under the ocean, in real deep parts of the ocean, right next to the crust plate boundaries. There is all kinds of seismic activity. There are bacteria there. There are bacteria in the Arctic, bacteria in the Antarctic. You look on this planet and you will find bacteria. It's the kind of thing that, we have sample systems that have gone to Mars and basically have little culture tubes, put material into the culture tube and wait for something to happen. Nothing happens. There is no place on this here planet that you could land some kind of rover, scoop up something, and not see bacteria. In the middle of the desert, bacteria. I mean, there are bacteria everywhere, which is really quite the testimony to just how flexible these little fellows are. They can do and grow in absolutely, ridiculously inhospitable conditions. And they've got some phenomenally weird ways of doing things. I think particularly we start thinking about what we know about our physiology. We start looking at what bacteria do, and there's some like, damn, that is some weird stuff. It's like, we don't have systems that can come even close to being able to use what they use for carbon, or different kinds of places to dump electrons, all kinds of things. It's like, man, I can't do that. So the autotrophs are a class of bacteria that we can kind of divide that are using a non-carbon-based energy source. And that, right there, I'll just tell you, we can't do. You can get bacteria that actually can consume things like iron and other salts as an energy source. Because, just to take a step back, if you think about what you had for breakfast today, you eat breakfast, or you eat in order to get energy, yeah? Yeah, okay. But you also eat to get carbon. Because let's face it, if you're eating stuff that's not carbon-based, you're eating some really weird things. I mean, don't eat glass, don't eat nails, don't eat dirt. You got to eat carbon. And, don't eat oil, but the forms of carbon that we can consume, starch, proteins, fats, we're using the energy in those carbon-carbon bonds to drive our metabolic reactions, yes, but we're also using that carbon to build strong bodies 12 ways. So everybody on the planet, all organisms, have got to get carbon in to generate macromolecules. But not everybody needs to get carbon in as an actual energy source, which is a really kind of a weird thing. So you can have autotrophs that take in carbon dioxide, but they're not photosynthetic like plants are. They're taking in carbon dioxide, using that carbon to build molecules. Their energy source is coming from completely different kinds of things. I mean, this is a really weird kind of operation. Heterotrophs is what we are and what a lot of bacteria are, and that they're using carbon-based energy. So the sugars and the carbon-based materials they're bringing in, they're using it for the carbon and also for the energy in the bonds. Now, in terms of medical relevance, it really is a small number of bacteria that are responsible for infectious diseases. In fact, by a huge margin, most of the bacteria that we find on the planet could care less, can't grow on you, are non-pathogenic, perfectly happy. So bacteria really are our friends. It's just a few bad eggs, always in the crowd. So we talk about the prokaryote cells. Now, this is actually a very cool area of microbiology, but as the bottom sentence says, it ain't medically relevant, so really we're going to skip it. But the archaea, this is a split in prokaryotes that's been around for probably 20, 30 years now. Back, in the ancient days, whatever, when I had microbiology in the 1980s, we had prokaryotes, that was it. We now have a lot more genetic DNA-based evidence to say, hey, there's some differences between these different groups in the prokaryotes. And so the archaea, the archaebacteria, they are really odd guys, and they are primarily living in very extreme environments, and they are pretty much autotrophic. They have to get carbon in, but they're not using it for energy, which makes them so far away from you and I that it's like, no, there's no way they're going to cause any kind of difficulty in our bodies. And we've got things that are heat-loving, which actually are really cool guys. Bacteria that can grow in near boiling conditions. So our 37-degree incubator, which is based on our body temperature, you got to grow these things in, a near oven. We got bacteria that can grow in real high concentrations of salt, also very cool. We have methane producers, we call them methanogens. Often these guys, particularly the methanogens, oxygen is a poison to them. You have to grow them in an atmosphere that has no oxygen. Consequently, it's not really easy to grow them in the incubator as well. There are some that are acid-loving, acidophiles. Bacteria end up being found in these really strange places like the dump ponds from mine tailings. It'll have, in gold mining processing, they'll use a lot of sulfuric acid. There'll end up being bacteria there. So there are all kinds of odd guys. The different divisions of the archaea, we don't need to worry about them, but some very cool and weird bacteria here. It just sort of emphasizes the fact that we're talking about the most diverse group of organisms on the planet.
This is kind of a random question. What kind of bacteria would you find in Antarctica?
Well, there are cryogenic or cryophiles, and they're going to be very slow growing. But some of them are using sunlight. There's all kinds of stuff that you can use. I think that being able to grow in a cold environment isn't nearly as difficult as being able to grow in a warm environment. You can take the bacteria that you got on your body and you can put them in, outside. Below freezing, depending, some of them absolutely can keep on going. They're going to go really, really slowly. In fact, even the most uber cool, hyper evolved for living in the Antarctic is a real slow grower just because of the thermodynamics. But growing in cold isn't nearly as hard as growing in heat. I mean, and we can see that for ourselves, and then you can put stuff in the refrigerator, let it sit, and you will still get bacterial growth in that guy. But if you put stuff in your oven, and, let's be generous and keep the temperature at like really a moderate temperature, like 120, there's nothing going to grow in that. I mean, you just are not going to find the thermophiles anywhere in our standard environment. And 120 isn't all that hot. So the desert, be interesting to see what you could find out there. And some of this stuff, let's face it, I make it up. I often go, you mean to tell me, like in the middle of the Sahara, you're going to find bacteria in the sand? Really? Probably so, but I'm like, okay, I don't know. So, the main thing is no human pathogens. So it's a cool idea, but nah. If you decide you want to get into industrial microbiology, go for it. All right, this is just another sort of cool microbiology or evolutionary biology discussion point that we can in fact break these guys down. So we have bacteria are recognized as being the more primitive form of life that prior to complex eukaryotes, there were bacteria on the planet. And bacteria then aid and evolve into the eukaryotes. And so we've got some real interesting steps here. The archaea are kind of along this route going into the eukaryotes. They were around long enough. You have protists, things that you may not think about, but like, the diseases that cause trichinosis, or the agents that cause malaria, Giardia. I don't know if anybody's ever had Giardia. It is a intestinal parasite you can get from drinking water that has not been treated. It's a common backpackers issue now because there's lots of different Giardia is kind of all over in the wild now. We got plants coming out here, fungi in terms of disease-causing agents, and we'll talk a little bit about fungi at the end. But you get yeast that can have yeast infections like thrush, athlete's foot, jock itch, all these fun things. In the Animalia, there are flukes and worms, so you can get various parasites like that. But I don't worry about them too much. Over here, amoebic dysentery is another kind of eukaryotic type disease. These are typically known as protists, these guys. It's what you find like in pond water and stuff. So things like amoeba and paramecium and things like that. All right, so then over here we have the bacteria as opposed to the archaea. And these are all prokaryotes, but this is where we find some of the things that you may be familiar with. So the more primitive guys, probably not so much. Photosynthetic organisms, what we used to think of as photosynthetic algae, but they are in fact bacteria. They don't have a nuclear membrane. And then we've got these different kinds of bacterial classifications of proteobacteria. These are what are referred to as gram-negative. We'll talk a little bit about that terminology in a bit. These are all kinds of things like E. coli, which is often found in ground meat and causes all sorts of food poisoning or death. Plague, dysentery, a whole range of different kinds of diseases in there. Syphilis, Lyme disease caused by spirochetes. This is actually probably should be over here. We have, actually the gram-positives. We have staph, strep, botulism. So there are really just a few spots in this tree that are pathogenic bacteria. One of the things that is happening is that there's all these various sort of notes and annotations and a work in progress. Is that when we get in the lab, we'll see that we can distinguish between bacteria pretty easily by selective media. So it's pretty easy to distinguish these different bacteria. Trying to decide where they fit on an evolutionary tree becomes a pretty tricky thing. You're trying to decide what is the more primitive characteristics and what are the more derived characteristics. And oftentimes, people will make some guesses and assumptions that are not necessarily correct. And one of the really cool things that the guys in my lab have been doing is cracking open bacteria and actually sequencing a bit of their DNA. And from there, we actually can get at some really cool evolutionary relationships, and we can identify pretty much any bacteria that you even think of. And I don't think that at a hospital, certainly not around here, they'd be doing that kind of bacterial identification. It's pretty easy to do, but it does require a little bit of equipment. But it is the ultimate way to identify different bacteria, and that is by looking at their DNA sequence. So, this is some basic cell biology and evolutionary biology stuff, but interesting to think about here. And that there's the notion that eukaryotic cells were derived from prokaryotic cells, in part because some of the organelles that we've got in our bodies, in our cells, like mitochondria and our plant friends with chloroplasts, these organelles have similarities to prokaryotes. And so this is known as the endosymbiotic theory, in that we have this very ancestral prokaryote, probably an archaea, because life back several billion years ago was pretty damn tough. Lots of salt in the water, not much oxygen, a pretty harsh place, real high UV light. We start getting membranes folding in, and you begin to generate a more organized kind of cell system. At some point, somebody engulfed an aerobic heterotrophic prokaryote. And we'll see is rickettsia and rickettsia-like bacteria are very odd bacteria, and they can't grow on their own. They actually have to grow inside another cell. And so there's pretty cool evidence that this might have actually happened, that these rickettsia-type guys sort of co-evolved, and you end up with somebody that decide, hey, I'm going to hang here. And this becomes the mitochondria. And the engulfing of a photosynthetic microorganism becomes a chloroplast. So one of the things we know is that from evolutionary time, when life begins, a couple billion years ago, the Earth is 3 billion, life's probably around 1 billion. We start seeing signs of life about 1 billion years later. The first life was absolutely bacterial, prokaryotic type stuff. And there came a point fairly quickly where photosynthetic organisms evolved, photosynthetic bacteria. And they did a particularly cool thing, and they began sucking up carbon dioxide. Going to use that as a carbon source. They're using sunlight as an energy source, but as we'll see when we get into metabolism, another thing you need to have is an electron source, because you can start making bonds, and those bonds need extra electrons. They get electrons from water, and in doing so, they generate a molecule of oxygen. So you have to take 2 molecules of water to get one O2, but they're doing it. And so another billion years goes by, and these guys have taken the atmosphere that was almost entirely carbon dioxide and nitrogen. They're pulling out a ton of carbon dioxide and spitting out oxygen from the water. They're splitting the water. And as the oxygen content starts to go up and up and up, because right now we're at about 18 to 20% oxygen. As the oxygen content comes up, you have this material, oxygen, that allows for much greater energy extraction from food. It's like this idea that you can't set a fire unless you have oxygen. And if you have a reduced oxygen atmosphere, you're going to have a pretty pissy fire. It's not going to be a really big fire. I mean, the way you stop fires is suck the oxygen from the room. I mean, I'm just starting thinking about this, like on a spaceship, if it had really low oxygen concentration, really low pressure, a fire is going to be just a really feeble thing. I mean, maybe get a spark, but you can't really get it to go. So, in order to have complex life, you've got to have oxygen. And absolutely, as the environment starts to get to have a higher concentration of oxygen, bam, here comes the multicellular organisms. Because without oxygen, you spend so much time just extracting energy from glucose in a really inefficient way that you don't have the ability to create multicellular organisms. I mean, I can't live without oxygen. Anyway, those are some of the things that, from a non-pathological perspective, I think are pretty cool to think about. All right, so this is another one of those things that is probably a useful list just to write out. But some of the different bacteria classes that are part of the disease pathology, and some that are just notable, like cyanobacterium, the what used to be called blue-green algae, but they are in fact bacteria. And just as an old question, how would you decide if a cell is a prokaryote or if it's a eukaryote? You say that because we know that algae are all eukaryotes, and you find this solution, this culture of free-floating cells that are able to carry out photosynthesis, and we call them blue-green algae, and they're fairly complex-looking guys, but if you use your microscope, you would see, they don't have a nuclear envelope, they cannot be eukaryotes. So that's one of the key things, is you look for the presence of a nucleus. All right, but we got the proteobacteria, and these are the kind of bacteria that we carry around with us all the time. So E. coli and various kinds of nearly E. coli, a lot of different subdivisions, and things like salmonella, a great food poisoning, Klebsiella, another good food poisoning. Yersinia pestis is what causes the plague. So we got some definite disease causers here. The firmicutes are a gram-positive. They've got a really rigid cell wall, and that just provides it. One of the things it does is it provides you with an easy target for antibiotics, because if you can block cell wall production, we ain't got no cell walls. You can kill those guys in their tracks. Staph, bacillus, which can cause anthrax, strep, Clostridium botulinum. This is where botulism comes from. Botulism is a whole lot rarer than I ever thought it was. Nobody gets botulism anymore, which is probably good, but from a disease perspective, it's a very cool disease. A couple of really weird guys, the spirochetes. These are sort of strange-looking bacteria because their shape is so weird, they're actually pretty easy to distinguish. And these are causing things like Lyme disease and syphilis. The chlamydia, these are these guys that can only grow in cells. They can produce sort of like a spore that can grow outside of a cell. And they are responsible for things like chlamydia, the STD. Actinobacteria, and these were actually initially thought to be fungi. And when you start looking at them more carefully, nope, they're bacteria. They're primarily in the soil, so you can go out there and you can find a whole range of actinobacteria. As it says, not particularly pathogenic, but they are a very cool source of a whole range of antibiotics. And they do this because, there's a limited amount of space in that little blob of soil, and if you can kind of push some guys out to keep your family up and running, you do. I isolated DNA from bacteria from soil. We probably will even do that in the lab. And even on a cold acid day, a spoonful of soil will have a ton of bacteria in it. I mean, you have to dilute it way out to get individual colonies. And you can find all sorts of different bacteria present in the soil. Antibiotics come from, a lot anyway, different kinds of soil bacteria. And so everything from ampicillin and penicillin is a mold, but let's see. I'm trying to think of some of them. Erythromycin, vancomycin. A lot of these guys are coming from other bacteria, streptomycin. So it is a constant source for identifying new sorts of antibiotics just by looking around in the dirt. we were doing some stuff trying to find some bacteria that would secrete antibiotics, and there was an article we read about some bacterium that was isolated from soil in Iraq was like producing a brand new, never before seen antibiotic. So one of the issues that we'll see when we start getting in the lab is that bacteria, A, they're small, B, they all look alike. I can't tell the difference. We have a lot of tests, but if you look at them under the microscope, and it takes a little bit of fiddling to get them under the microscope to be able to see them, because they are pretty much at the limit of light microscope. If they were much smaller, you really couldn't see them with a light microscope. But with some care, we can see them. And we've got these old-fashioned terms that are used to kind of describe the shape. So the term coccus is a sphere. And so we've got diplococci, Neisseria, which is sort of an odd shape, but tetrads and sarcinae, cocci packets of multiples of 8. So terms you probably have heard of, streptococcus, and this is a chain of spheres. Staphylococcus is not particularly a chain, but it's sort of an unorganized mass, a blob. And then there are bacillus shape, or the bacillus class. So the names of some of the bacteria reflect the Latin term for shape. I assume bacillus means rod in Latin, I don't know. So bacillus are definitely not spherical. They're a little bit longer out. And so we have then kind of the addition, streptococcus mean chain cocci. Streptobacillus are chain bacillus shapes. Let's see. Streptomycetes, these are forming these little filaments. Another major class is these spirally guys, or curved guys. They have sort of like a comma shape to them. And so things like spirilla and spirochetes, which are like Lyme disease, Vibrio, and I think Vibrio causes different kinds of choleras, I think. Regardless, it's a kind of a comma-shaped bacterium. And so a lot can be told by looking at the shape of the bacterium as to what the individual species is. So we've got a whole range of tests. And we're going to start next week in lab on Wednesday at 1. Just let me know, and I'll remind you again on Monday and Wednesday. And we're going to just start this coming week with doing some basic culturing techniques. But pretty soon we'll get into using the microscopes and stuff. And so shapes are a really good way of identifying sort of the base identification. Being able to look at structures, cell wall type, if it's there or not. There are things like flagella, the endospores, which are present in some and absent in others. Those can be used as a way of keying out the individual species. The RNA sequence, ribosomes, is what we do in the lab a lot. That is what requires a little bit more technology. There are a whole bunch of different stains that we can use that will react to the different parts of the cell wall. Some will stain in different ways. Metabolism is a biggie. By looking at the requirements for growth, we can begin to identify different bacteria. There are going to be bacteria that can break down fats. There are bacteria that can break down one type of sugar but not another type of sugar. There are bacteria that can generate acids. There are bacteria that grow in acids. And by looking at these different kinds of metabolic tests, basically asking, do you grow on this or do you not grow on this, we can begin to start to narrow down what the individual bacteria is. A cool thing, I don't think we're going to be doing this, but we've got, it's a company called Biolog that I think may be actually used in the hospitals that you can use to identify bacteria. I personally would rather crack them open and sequence them. But Biolog has got like a 96 well plate filled with a bunch of different sugars in it, all these different kinds of metabolites. And you put your bacteria in all those things, And then you look to see if you can break them down. And there's a dye indicator that shows if you're actually liberating electrons, that is breaking bonds. And by looking at the sort of matrix of combinations of, yep, you can break down this one and this one and this one, but not these two, you can begin to identify what the bacteria is. And so we'll be doing that sort of on a larger scale, looking at plates and things and see how they grow. Let's see, another mechanism is to use antibodies, serology. And this is what's happening when you go to the doctor and they do that really rapid strep test. I mean, have you guys ever had that done on you where they stick the swab in the back of your throat to make you gag and your throat is sore anyway? And then you don't see it, but what they're doing is taking something that looks very much like a pregnancy test kit thing. You take the swab, mix it in a saline solution, vortex it up, and then put a drop of it on a little dot. The dot is, in fact, a spongy wick. It pulls the solution through, and there's an antibody that's in the part over here, bound in such a way that if it binds to the molecule or the bacteria or whatever you're looking for, you'll get a red line. And if it doesn't, you won't get a reaction. And then there's a little control that's always going to turn red that lets you know, okay, this works. And so this is like a standard antibody strip kit that is used for tons of things. Pretty much if you can make an antibody against something, we'll be talking a little bit antibodies later, you can build one of these strip kits. And so a standard thing to do is looking for blood in your poop. It does require that you get a little poop sample, which is, I'll just leave that to your imagination. But you can take this little brush, once you've stuck it into your turd, put it in saline, take a drop, put it on there, and in 5 minutes, you know if you've got blood in your stool, which if you did, it's probably not a good thing. You should have a colonoscopy. You can look to see if you're pregnant by the presence of specific hormones that are going to be found in your urine. You can begin to see different kinds of bacterial infections because individual bacteria are going to have different kinds of sugars and things on their cell surface. What I am waiting for, and I'm beginning to hear little stories about this, is a COVID-19 strip test. We ain't got it yet. Part of it is that COVID-19 being a coronavirus, it's difficult to get a real specific COVID-19 positive test versus just one that's testing for coronaviruses. But I personally would be just fine with that. It's like, okay, go ahead and spit in here, and now while you wait, you got a red line or are you clear? Oh, you're clear, come on in. You got a red line? Get the hell out of here. I don't know that you have COVID-19, but you got some coronavirus, and I don't want to talk to you. And don't sneeze on me, and there's the door. So we're not doing that yet. That would be nice, because, like everybody getting on an airplane, spit. Oh, you're good. Get on. All right, so DNA sequencing is kind of the end-all. That pretty much is the way you can begin to identify individual species. Not foolproof, damn close to foolproof. There are some things that can squirrel it out, and you can pick up different kinds of sequences and mutations and stuff. So the ultimate goal of this is to develop what we call a dichotomous key, which I'm not sure if you guys have seen anything like this yet or not. And we'll be playing with them in lab. A dichotomous key is a stepwise yes-no path to identifying an individual organism. And so you ask a series of questions, like, first off, is it gram-positive or is it gram-negative? Gram stain is a stain that's going to ask whether you have a cell wall or not. If you have a cell wall, you exclude one of the colors. If you don't have a cell wall, you let that color in. When you wash it, you'll either be kind of a pinky or you'll be a dark violet. All right, so if you're gram-negative, you go to step 6. And we're going to ask, do you have one flagella visible or no visible flagella? If you're gram-negative and no visible flagella, it's probably an E. coli. If you have a flagella, you got to go here. And do you have more than one? Okay. You're one of these two. If you only have one, We have a case of cholera on our hands. Oh, great. And so you can work your way through these different steps, and this is known as a dichotomous key. So, if you're gram-positive, gram-negative, if it's gram-positive, you go here. If it's a bacillus, then we know it's going to be, probably one of these two guys, and we can look to see if they have spores, all kinds of different tests. And so, depending on where the path takes you, we have all kinds of tests that we can carry out. And so what we're going to do is A, look at a variety of different tests, B, give you unknowns that are based on the tests we have. We're not going to give you anything that's like, I need to do this test. We don't do that. No. And happily, in terms of medically relevant bacteria, there just aren't that many. I mean, you may find some oddballs. Sure, somebody comes back from New Zealand or Australia and has some sort of weird-ass stuff. But for the most part, the kinds of bacteria you're going to see are just really going to be a handful of them. So identifying it's important. All right, just lastly, we talked a little bit about light microscopy versus electron microscopy, but it's worth throwing out here. We're going to be using some light microscopes a bit in the lab. We got some pretty nice ones now. But light microscopes are limited in their resolution. Light microscopes are very much like, low resolution. And you're limited by the wavelength of light. And it actually becomes some fairly esoteric physics weirdness. Blue light, being a shorter wavelength, has better resolution than red light. And so to push the limits of light microscopy, you can use blue light, and that can help a little bit, but not all that much. If you really want to increase the resolution of light of microscopes, you got to go to something that's got a smaller wavelength, and that is an electron. And so with a light microscope, you have, there are multiple types, but we're going to be using a standard compound microscope in which you have the light source at the bottom coming up, and then a series of lenses that are focusing the light, and then an objective, which is essentially the big magnifier at the base, and then that comes up, and then you have optics at the top, which are sort of a smaller magnification, and that's how we can begin to magnify different kinds of samples. There are a variety of different kinds of polarizing light systems where you can polarize light, where you have light that is not being scattered. And you can look for, since you know that you're putting light in, sort of like polarized sunglasses, in one direction, and then when it hits the sample, it will scatter, and you can look and see that kind of scatter effect, which will make it look a little bit like shadows and things. So you can get some really cool images that way. But light microscopy is limited. The electron microscope is a different kind of game in that you actually are beaming from the top, where you have basically a gigantic circuit. The anode is dropping electrons. This whole thing has to be kept into a vacuum. The electrons, if they hit air, will scatter. So you got to pull a really hard vacuum in this thing, and then you shoot electrons down, and your sample is down here. There are electromagnetic lenses, essentially. You don't really have glass. You have ways of changing the shape of the electron beam with electromagnets. That allows you to actually change the magnification. And then you end up shooting the electrons through your specimen. The electrons will bounce off and not pass through something that's got heavy metals and stuff in it. And that'll end up being sort of a black spot. And then you have a fluorescence screen that is kind of like an old-style TV screen. The electrons hit it, they light up a little pixel. And you can get all kinds of really high-definition imaging here. The real problem is that with light microscopes, you can take living samples. So you can take a culture off the plate, spread it kind of thin on a glass slide, put it under the scope, and you can see it. With electron microscopes, you have to have these things in a vacuum. That means they have to be completely dehydrated, so you pull all the water out. The standard electron microscope like this is what's called a transmission electron microscope. The samples have to be so thin, they're actually thinner than a single cell. And in order to get slices that thin, you have to put the entire sample into epoxy. And so you mix up a little bit of epoxy, put your sample in there at the tip, and then you have what's called ultramicrotome, which is a device that takes very, very thin sections. And if you've been in A&P, you maybe have seen light microscope sections for looking at tissues and stuff. Those are usually done with paraffin. This is done with epoxy. And then you take the sample, put it on a little copper grid, looks like the size of a back end of a pencil eraser, and you then take these grids and soak them in some phenomenally nasty chemicals, uranium salts, and different kinds of lead salts. And what that's doing is interacting with some of the compounds and the lipids and stuff in the cell. And you generate a sample that's got these really heavy metals associated with different parts of it. The electrons hit that, they can't go through, and that's what gives you this sort of cool black and white image. And what it means is that the samples are really, really dead, and they have to be really thin. And so there are things that are hard to actually really understand, like big structures, because you're looking at teeny little sections of them. So another cute trick that we can use is have an antibody that is coupled to a fluorescent molecule. And this is a form of light microscopy known as fluorescence microscopy. A couple of questions and things, but nothing too major here. We don't have an electron microscope on campus, which is probably, if they're a pain in the ass to use. So let's see. Lastly, the idea was that bacteria are single cells and they live life as a single cell. We, of course, are multicellular organisms, and individual cells are always communicating with each other through hormones, through cell-cell connections, all kinds of stuff. Bacteria are able to actually coordinate activities. So, the original idea is that only in multicellular organisms, A, do you need a method for cellular communication, and B, can you actually have a method? Does it actually happen? We figure that bacteria are just dots, and they act as dots. So we now know that pretty much all cells have the ability to coordinate activities. And this has got some pretty significant consequences. Primarily, you can get bacteria that actually can send out signals that this is a good place to hang out. And bacteria then, their brethren and sisters can begin moving around and crawling to form what's called a biofilm. And this can coordinate these various sorts of activities of beginning to break stuff down. This is becoming a really critical problem in things like transplant surgery, or not transplant surgery, joint replacement surgery. Like hip joints are a classic place where biofilms can occur. I'm not sure if you've ever seen a hip replacement, but it looks like a trailer hitch attached to about a foot-long, massive spike. And you pop off the top of the tibia, the actual ball, and you carve out a hole, and you insert this trailer hitch in there, and that's going to work. Okay, great. But you can end up, potentially, with a little bit of bacteria in there that starts to actually coordinate its bacterial growth and begins to form these biofilms, and it can be incredibly difficult to remove it by antibiotics, to the point that sometimes they actually have to go back in, pull that implant out, swab the whole area out, and try again. Bad news. Anyway, there is this thing that is called quorum sensing, and what it can do is end up bringing a lot of bacteria into one area. So there you go. These staphylococcus films on catheters, implants in things in bone. It's the standard sort of thing that if you have a catheter shoved up yourself to allow you to pee into a bag, lovely. I don't think I've ever had one, but we don't even think about it. But having a small silicone tube shoved up your pee hole, great. You let that sit for a while, and it begins to accumulate bacteria. I mean, typically, any kind of hole that you have in your body is a spot for bacteria to get in. Respiration system is a great place, but pretty much all your holes are possibilities. And with the urethra, you're constantly flushing stuff out as you pee. So peeing is always good. You put this tube in there, you're no longer flushing out the system, you're flushing it through the tube, and it is pretty much not a will it happen, but when will it happen, and that you're going to get some kind of urinary tract infection by having a catheter in there. So let's do our best to not have catheters. All right, I'll catch up with you on Monday. We're going to start getting into some actual microbiology specific, and